Good day, dear students. We are beginning our biochemistry lecture, and today it will be devoted to integration of metabolisms of carbohydrates, lipids, and proteins. Please write the topic in your notebook. Integration of metabolisms of carbohydrates, lipids, and proteins. In previous lectures, you studied catabolism, anabolism of carbohydrates, lipids. In next semester, you will study biosynthesis and catabolism of proteins, and you can decide that these are completely different processes in our organism, but in reality this is one large metabolism with many connections, and today I try to show you all connections between all these processes, and I hope that after this lecture you will see that we have large mutual metabolism of all these three important groups of biomolecules. You need for our scheme, which we will create today, the whole empty page in your notebook, and I will use the blackboard. So if you have a page on which you already wrote something, please go to another empty page for creation of our scheme. So let's begin, and we will begin with metabolism of carbohydrates. Let's decide that our blackboard is divided into three parts. The left part is blood, central part is cytoplasm of the cell, and the right part of our blackboard and also of your page in your notebook is the mitochondrion. And first of all, we will begin with glucose. So let's leave empty three lines on the top. And from the very left side, on the fourth line, let's write glucose. Glucose. So glucose can appear in our blood from food or after the degradation of glycogen in our tissues. But finally, we are receiving glucose in our blood, and it must penetrate through the cell membrane inside of the cell to cytoplasm. So let's make such drawing. This is the double membrane of the cell, and first step is the penetration of glucose inside to the cytoplasm. So glucose out of cell, and now glucose inside. But you know very well that glucose can be transformed only after appearance of glucose 6-phosphate. So the first very important regulative reaction is transformation of glucose to glucose 6-phosphate. Let's show this. Glucose 6-phosphate. And we have two very important enzymes for this reaction. In muscles and other tissues, we have hexokinase, while in liver for this reaction we have glucokinase. You know this. this reaction is irreversible. So under the influence of these enzymes, glucose can be transformed to glucose 6-phosphate, but we cannot receive back reaction with these enzymes, glucose 6-phosphate for glucose. For this, we have another special enzyme, and we shall tell about this a little bit later. Now we have glucose 6-phosphate, and with this substance we can obtain two ways. First way, when we have much energy in our organism, it can be transformed to polymer to glycogen. When we have no energy, glucose 6-phosphate go to glycolysis, then to citric acid cycle, and we are receiving energy. So let's begin with glycogen. So on the top of your page and on the top of my blackboard, I am writing glycogen. You know that glycogen is a polymer of glucose. So glucose at first is transforming to glucose 1-phosphate, and then after polymerization we are receiving glycogen. I want to say you that we will not write all stages, all names of molecules, because we have no enough place in your page, in my blackboard, on my blackboard. And we shall write only the main steps of transformations. So glucose 6-phosphate after glucose 1-phosphate can be transformed to glycogen, and for this we have special enzyme glycogen synthase. Glycogen synthase. For degradation of glycogen, we have another enzyme, and you also know its name. It is phosphorylase. Phosphorylase. The activity of phosphorylase depends from the influence of two important hormones, epinephrine and glucagon. When we need energy, our organism produces epinephrine and glucagon, and as a result we have activation of phosphorylase, and glycogen will be transformed to glucose 6-phosphate with the following glycolysis and receiving of energy. So this on the top here, let's write epinephrine, and you know, in the pancreas we have two enzymes, insulin and glucagon. So the second one is glucagon. These two hormones activate phosphorylase, and not only phosphorylase but also lipase, but this we shall tell a little bit later. So now we have the top of our scheme, and let's go down with glycolysis. Glucose 6-phosphate can be transformed to fructose 6-phosphate, This is the simple reaction of isomerization, but the next is very important. Fructose 6-phosphate will produce fructose 1,6-bisphosphate. fructose 1,6-bisphosphate. And we have very important regulative enzyme for this reaction. We call it phosphorylase. Phosphorylase. Then molecule of fructose 1,6-biphosphate will be divided into parts, and finally we shall receive dihydroxyacetone phosphate. Dihydroxyacetone phosphate. After a few transformations, we are receiving another substance, phosphoenol pyruvate. Phosphoenol pyruvate. So I shall stand here and you will see phosphoenol pyruvate. And the next reaction will be also important and regulative. This is transformation of phosphoenol pyruvate to pyruvate. For this we have important enzyme, pyruvate kinase. And this is the last reaction of aerobic glycolysis. So then we will go to citric acid cycle, but at first let's discuss some things connected with glycolysis. Many reactions of glycolysis are reversible. It means that with the same enzyme, a reaction can go to both directions. But we have three exceptions. Irreversible reactions are reaction with hexokinase or glucokinase, with this is not phosphorylase, but phosphofructokinase. Please correct. Phosphofructokinase. Phosphofructokinase, please write here, phosphofructokinase. Phosphofructokinase is the main regulative enzyme of glycolysis. And this reaction is also irreversible. And the third irreversible reaction is transformation of phosphoenol pyruvate to pyruvate. Reaction with the enzyme pyruvate kinase. What to do if we want to go back from pyruvate to glucose? We have problems with these three reactions, with hexokinase reaction, phosphofructokinase reaction, and pyruvate kinase reactions. But we have special ways, and now we shall discuss how these reactions can be go back. Glucose 6-phosphate can be transformed to glucose, but not with enzyme hexokinase or glucokinase. For this we have different enzyme, glucose 6-phosphatase. So it is different from hexokinase. So reaction is irreversible because it can go only to one direction, only in one direction with hexokinase and glucokinase. For this reaction, we need another enzyme, and this enzyme is glucose 6-phosphatase. Another reaction with another enzyme, we have for transformation of fructose 1,6-biphosphate to fructose 6-phosphate. And this is fructose bisphosphatase. Let's write the names of these enzymes. Here we have glucose 6-phosphatase, and here we have fructose 1,6-biphosphatase. Bisphosphatase. Fructose 1,6-bisphosphatase. Here. For transformation of pyruvate to phosphoenol pyruvate, we have no such simple solution. For this, we need transformation of pyruvate at first to oxaloacetate. And we shall discuss this, but at first we must write the beginning of the citric acid cycle. And as you know, pyruvate must go to mitochondrion. So let's write first a long arrow and show that pyruvate now is inside of mitochondria. Here we have, you know this very good, pyruvate dehydrogenase reaction, and we are receiving acetyl coenzyme A. Acetyl coenzyme A. Then acetyl coenzyme A reacts with oxaloacetate. So let's write. And now we have the beginning of citric acid cycle. But before beginning of citric acid cycle, let's discuss how pyruvate can be transformed to phosphoenol pyruvate. For this, we have two stages. The first stage is pyruvate with the enzyme pyruvate carboxylase. Let's write this. Pyruvate carboxylase can be transformed to oxaloacetate. Here we must write somewhere carboxylase. Here. Pyruvate carboxylase, and we are receiving oxaloacetate after addition of carboxyl group to pyruvate. Then oxaloacetate can be transformed to phosphoenol pyruvate. The next arrow will go here. And for this reaction we have another enzyme, phosphoenol carboxykinase. Let's write carboxykinase. So look, we have here another small cycle which is consisting of three substances: phosphoenol pyruvate, pyruvate, oxaloacetate, and back to phosphoenol pyruvate. Let's repeat these three enzymes which are important for this process. Phosphoenol pyruvate goes to pyruvate with pyruvate kinase. Then pyruvate goes to oxaloacetate with pyruvate carboxylase. then oxaloacetate goes to phosphoenol pyruvate with phosphoenol pyruvate carboxykinase. So we have such small cycle, and this is the reaction of gluconeogenesis, you know this. So if our organism will decide to produce glucose from pyruvate, the way will be as follows. Let's look. Pyruvate will be transformed to oxaloacetate, then oxaloacetate to phosphoenol pyruvate. This is because this reaction is irreversible. Then reversible reactions to fructose 1,6-biphosphate. Reaction with phosphofructokinase is irreversible, so we need fructose 1,6-biphosphate, biphosphatase enzyme, fructose 1,6-biphosphatase. Then we are receiving fructose 6-phosphate, then glucose 6-phosphate. This hexokinase reaction is also reversible, so we need another enzyme, glucose 6-phosphatase, and finally we are receiving glucose. So three irreversible reactions: reaction with hexokinase, glucokinase first, with phosphofructokinase second, with pyruvate kinase third, and for the way back we need, instead of these three enzymes, four another enzymes: pyruvate carboxylase, phosphoenol carboxykinase, then fructose 1,6-biphosphatase, and glucose 6-phosphatase. This is information that I remind you, and this is connected with gluconeogenesis. This process we will need during our lecture today many times, so I think that it is important that I repeat this few times. Then let's go to citric acid cycle. I hope you know it very good. After reaction of acetyl coenzyme A with oxaloacetate, we are receiving citric acid, citrate, then isocitrate, then alpha-ketagluteric acid, alpha-ketaglutarate, then after alpha-ketaglutarate we have succinyl coenzyme A, then succinate, then fumarate, malate, and back to oxaloacetate. Let's connect all these substances with arrows, and we will receive citric acid cycle: oxaloacetate, citrate, isocitrate, alpha-ketaglutarate, succinyl coenzyme A, succinate, fumarate, malate, oxaloacetate. This is citric acid cycle. Citric acid cycle. So this is more or less all with metabolism of carbohydrates, but at the end of this part of our lecture, I must tell you about one very important irreversible reaction. It is a reaction of transformation of pyruvate to acetyl coenzyme A. You know that pyruvate dehydrogenase complex is the enzyme complex of this reaction, and this reaction is absolutely irreversible. if here we have different enzymes or different ways to return, in this case we have no such possibility. So this reaction is absolutely irreversible. And if we have problems with citric acid cycle and a lot of acetyl coenzyme A appear in our organism, there is only one way, by synthesis of so-called ketone bodies. So ketone bodies. You know, ketone bodies are three substances which can be produced with acetyl coenzyme A. This is not a problem for a healthy person that this reaction is irreversible, but when the patient has some diseases, for example diabetes mellitus, this will be a large problem, because all acetyl coenzyme A will not go to citric acid cycle but will produce ketone bodies, and it can cause different symptoms. Now we must connect these reactions of carbohydrate metabolism with lipids and with proteins. Let's begin with lipids, for example with fats. So fats, you know that fats consist of glycerol and fatty acids. And for degradation of fats, we need enzyme lipase. This lipase we have in our intestine, and also we have in our tissues. This is a little bit different structure of this enzyme, but they cause the same reaction, degradation of fats to glycerol and fatty acids. Let's write this. Glycerol and fatty acids. What will happen with glycerol? We have here in glycolysis we have dihydroxyacetone phosphate, and after phosphorylation of glycerol it can be transformed directly to this dihydroxyacetone phosphate. So we can make such a simple arrow. For this transformation we need one molecule of ATP, but it doesn't matter. In our scheme we have very simple arrow from glycerol to dihydroxyacetone phosphate, and then dihydroxyacetone phosphate will produce pyruvate, or if it is gluconeogenesis, it will produce back glucose. And fatty acids. For fatty acids, you know, we have the process which we call beta oxidation, and the result of beta oxidation is the molecules of acetyl coenzyme A. So we must connect fatty acids with oxaloacetate. Let's go here. And here. I shall show you once more with the stick. So fatty acids, this arrow, and we are coming to acetyl coenzyme A. Then acetyl coenzyme A go to citric acid cycle and produce energy. As I told you, many reactions of glycolysis are reversible, except these three. So we can write back arrows in all reversible reactions. So this is irreversible, but this is reversible. Then this is reversible, this irreversible, and this is also reversible. So the way back, we shall decide to obtain glucose from pyruvate. The way back, I shall remind you, is like this: oxaloacetate, phosphoenol pyruvate, then simply back, then this reaction with fructose 1,6-bisphosphatase, then simply back, and this reaction with glucose 6-phosphatase, and we are receiving glucose. That's all with lipids, with fats. And let's discuss now catabolism of proteins. So we have some empty place here. So let's write proteins. But before beginning with proteins, I must show you one more arrow. As I told you, fats is producing glycerol and fatty acids, but under the influence of lipase. Let's write this. Lipase, because I told you this, but I think that it is important that you will have it in your notebook, because you do not copy all that I am speaking, but copy only that I am writing. This I know. So lipase is the enzyme that catalyzes the degradation of fats. And I told you already that epinephrine and glucagon are also activators of lipase. So look, these two hormones, epinephrine and glucagon, as I told you, we are producing when we need energy, and first they activate phosphorylase and degradation of glycogen for receiving of energy, and second they activate lipase, and we have activation of degradation of fats, and so also we are receiving energy via acetyl coenzyme A, and we are dihydroxyacetone phosphate. Now let's go to proteins. You know that proteins are polymers of amino acids, and we have two types of amino acids if we decide to say about their metabolic ways. The first group of amino acids are glucogenic, and second is ketogenic amino acids. So first group, glucogenic, we call them glucogenic because they can produce glucose. And second group is ketogenic amino acids. We call ketogenic because they can produce ketone bodies. Now we must write all these arrows, and you will understand how it can be happen. So proteins, at first let's write ketogenic amino acids, and then glucogenic amino acids. Ketogenic amino acids, and glucogenic amino acids. We must immediately say that there are two amino acids, please write it somewhere separately, that there are two amino acids that can be in the same time ketogenic and glucogenic. This is tyrosine and phenylalanine. We shall study this in the next semester, that after degradation of these two amino acids, we are receiving fumarate, which comes to citric acid cycle, and acetacetic, which is producing finally acetylcoenzyme A. Acetoacetate, if to say correctly, and finally acetoacetate also producing acetylcoenzyme A. So phenylalanine and tyrosine are divided into parts. One part is glucogenic and second part is ketogenic. All other amino acids or ketogenic or glucogenic. And ketogenic amino acids will produce and can produce acetylcoenzyme A. Let's make this arrow. Look, these two arrows are parallel one to another. Fatty acids are producing acetylcoenzyme A, and ketogenic amino acids are also producing acetylcoenzyme A. So after acetylcoenzyme A, they can be transformed to ketone bodies, and so we call them ketogenic. Not so simple with glucogenic amino acids. Glucogenic amino acids can be transformed, different amino acids can be transformed to different molecules of this metabolism. Let's make some examples. For example, alanine and some other small amino acids can produce directly pyruvate. Look. So very short arrow from small glucogenic acids to pyruvate. If you take alanine, then after taking away amino acid and changing it to keto group, you will immediately receive pyruvate. If you remember the formula of alanine, it is very simple for you to understand. So changing of amino group for keto group in alanine, and we are receiving pyruvate. If we shall make the same transformation with aspartate, we are receiving oxaloacetate. So next arrow will go here, and we can obtain oxaloacetate from, once more I shall show you, these are arrows of glucogenic amino acids. Some can be transformed to pyruvate, some to oxaloacetate, for example aspartate. If we shall take glutamate and make the same procedure, we immediately receive alpha ketoglutarate. So another arrow will go here, and some amino acids can produce succinyl coenzyme A, and as I told you, two amino acids, tyrosine and phenylalanine, can produce fumarate. And finally, we have all arrows. Let's check once more. So some glucogenic amino acids will produce pyruvate, some alpha ketoglutarate, some succinyl coenzyme A, some succinate, and some oxaloacetate. And now let's solve some examples. For example, if we have aspartate. Aspartate is glucogenic amino acid. So on our scheme, we can simply show how aspartate can produce glucose, can be transformed to glucose. Let's look at our scheme. So after degradation of protein, we are receiving aspartate, which is glucogenic amino acid. I repeat, this is example. Then aspartate after transamination, so changes after change of amino acid and amino group and keta group, it will produce an oxaloacetate, and then oxaloacetate, let's go with this arrow, with phosphoenol carboxykinase will produce phosphoenol pyruvate. Then I'm going here, and then other things we already know, that this reversible reaction, this reaction irreversible, so we have this bridge with fructose 1,6 bisphosphatase. Then this reaction is reversible, then another bridge with glucose 6 phosphatase, and then we are receiving glucose. So after this we can simply say, oh yes, aspartate is glucogenic amino acid, because with this way, once more I'm showing you, I'm showing this arrow, this, we are receiving glucose. Let's take another example of what we should take. For example, glutamate. Let's take glutamate. Glutamate is also glucogenic amino acid. So we are going here, and after transamination, we are receiving this arrow, alpha ketoglutarate. Then we need to go to citric acid cycle. So alpha ketoglutarate will produce succinyl coenzyme A, then succinate, then fumarate, then malate, and finally we are receiving once more oxaloacetate. After this we know the way. Oxaloacetate, phosphoenol pyruvate, way back, and glucose. So all glucogenic amino acids finally have the way to glucose, so we call them glucogenic. And once more, all ketogenic amino acids are producing acetylcoenzyme A, because they can be transformed to acetate or acetoacetate. And finally, acetylcoenzyme A. And from acetylcoenzyme A, we can obtain ketone bodies. And I can remind you that two amino acids, phenylalanine and tyrosine, are divided in two parts. One part is producing glucose. This is via fumarate, and second part is producing acetylcoenzyme A via acetoacetic acid, acetoacetate, and produce ketone bodies. I hope that now you have a lot of information, and all these things that I show you on the blackboard we have in the healthy organism. And now we will discuss what will happen at pathology. And as example, we shall take diabetes mellitus. Diabetes mellitus. I shall write here on the top diabetes mellitus. If we shall speak in Latin language, not in English, it is diabetes mellitus. Diabetes mellitus. In English, diabetes mellitus. You need to take fountain pen with another color, for example, with red ink or green ink, and with this different color, we shall show what will happen when the patient has this disease, diabetes mellitus. Scientifically, this disease was described in the first century by two Roman physicians, and they gave this name diabetes because the patient is producing a lot of urine, a lot of water, and the translation of the word diabetes from the Greek language is producing of a lot of fluid. So it means that this patient is producing a lot of urine, and at that time it was a very dangerous symptom, because there was no idea how to treat this disease. Only in 17th century scientists understood that this disease is also connected with the increasing of the concentration of glucose in urine and in blood serum, and this disease obtained second name in its name. It is mellitus. It means you know that mellitus it is something that it is in English it is honey diabetes. Mellitus, it is what bees are producing. So in Russian we are saying sugar diabetes, but in Latin it is honey diabetes, diabetes mellitus. And only in the end of the 19th century scientists discovered that this disease is connected with pancreas. It was 1889, we know the date, where scientists understood what happened in the organism, that this is the disease of pancreas. But it was not possible to treat this disease until 1922. I must find somewhere the empty place. Let's write here. In 1922, a Canadian scientist, Frederick Banting, obtained pure solution of the hormone insulin, and after these injections of insulin can save a lot of lives of people with diabetes mellitus. In 1923, Banting received Nobel Prize for this investigation and for this discovery. Before we are discussing, before we will begin to discuss what will happen in the case of diabetes mellitus, we must make some marks on our skin. So now we know that diabetes mellitus is the disease of patients when these people have no insulin or have low concentration of insulin, hormone of pancreas. So let's write here, insulin. In some cases, the concentration of insulin is not very low, but tissues does not react. Tissues do not react to the influence of insulin. This is also diabetes mellitus. So we have two different situations. But in both of them, insulin cannot show its function because of low concentration or because of tissues that do not react to the influence of insulin. Why insulin is so important? Because it influences to some enzymes of the processes that we have on our drawing. Let's write somewhere on the separate place a sentence that I tell you and then make some marks here. So in the presence of, let's write, in the presence of insulin, the biosynthesis of some enzymes, in the presence of insulin, the biosynthesis of some enzymes is possible. So we have insulin, we have biosynthesis of these enzymes, we have these enzymes. No, we have no insulin, biosynthesis does not happen, and we have no these enzymes. Now let's tell what enzymes. And not surprisingly, that these are enzymes that are most important and regulative, about which we already told many times. This hexokinase, glucokinase, phosphofructokinase, pyruvate kinase, it is here, pyruvate kinase. Then glycogen synthase, and insulin also influences on penetration of glucose through the cell membrane. So this is the place of the influence of insulin. Let's repeat once more. So, biosynthesis of some enzymes is possible only in the presence of insulin. What enzymes? Glucokinase, hexokinase, phosphofructokinase, pyruvate kinase, glycogen synthase, and insulin also influences on penetration of glucose through the cell membrane. Let's make this better if you mark with the different color, for example with red color, and then let's make some additional arrows. So insulin, then arrow to glycogen synthase, arrow to phosphofructokinase, arrow to pyruvate kinase, and arrow to glucokinase and hexokinase, and arrow to penetration of glucose through cell membrane. So many arrows. Now we know that if insulin cannot fulfill its function, we have no glycogen synthase, so the synthesis of glycogen is not possible. Let's cross here, also with red color. Then glucose cannot be transformed to glucose 6-phosphate. Second cross. Then glucose cannot penetrate through cell membrane. Fructose 6-phosphate cannot be transformed to fructose 1,6. Fructose 6-phosphate cannot be transformed to fructose 1,6-biphosphate. And phosphoenol pyruvate cannot be transformed to pyruvate. So 1, 2, 3, 4, and 5 blocks. In the case of diabetes mellitus, we also have problems with citric acid cycle. Why? Because for citric acid cycle we need enough concentration of oxaloacetate. But from where are we receiving oxaloacetate? One of the main ways is carboxylation of pyruvate. But look, as glucose cannot penetrate to the cell, then cannot be transformed to glucose 6-phosphate, then cannot be transformed to fructose 1,6-biphosphate, then cannot be transformed to pyruvate. Finally, we have a low concentration oxaloacetate, and citric acid cycle cannot work enough for producing energy. As a result, the patients of diabetes mellitus have some important symptoms. Let's now discuss symptoms of this disease. First symptom that was described, as I told you, more than 20 centuries ago, it's a lot of urine which produce the patient. We call this polyuria. Polyuria, so many urine, polyuria. Why? Because the organism of the patient needs energy. And this way is blocked. So patient tries to obtain energy from fats. But from fats, he is obtaining glycerol that can be as a result of gluconeogenesis produce glucose, because look, this reaction is blocked, but this not. This reaction is blocked, but this not. This reaction is blocked, but not this. So gluconeogenesis is possible. So the molecules of glycerol will produce glucose, and the molecules of fatty acids are producing acetyl coenzyme A and ketone bodies, and they cannot produce glucose. So the patient tries to obtain energy from proteins, and degradation of proteins will give him ketogenic amino acids, and finally acetyl coenzyme A and ketone bodies, and glucogenic amino acids. And remember, I told you that every glucogenic acid can produce through citric acid cycle, oxaloacetate or pyruvate, new and new molecules of glucose. And this high concentration of glucose in blood is not good for the human organism. So the patient needs to take away this glucose, and the main way is through kidneys with urine. So kidneys are producing much urine to take away glucose from the organism. What will happen later? The organism are losing a lot of fluid. So the patient needs to drink a lot of water, and a lot of urine, a lot of water for drinking, a lot of urine, a lot of water for drinking. And this second symptom, when the patient is drinking a lot of water, we call polygypsia. So polyuria causes polygypsia. When I'm asking students at examination why the patient with diabetes mellitus is drinking so many water, students immediately say, this is very simple, because these patients are producing a lot of urine. Okay, second question. Why are they producing so many urine? And immediate answer, because they are drinking a lot of water. But why they are drinking a lot of water? Immediate answer, because they are producing a lot of urine. Okay, they are producing a lot of urine. Why? Because they are drinking a lot of water. And no end of this discussion. You understand. You must know very good that the primary symptom is polyuria, producing a lot of urine, because organism must take away a high concentration of glucose. And polygypsia, drinking a lot of water, is a secondary symptom. Let's go further. Patients cannot obtain enough energy, because look, this 1, 2, 3, 4 blocks on this way, 1, 2, 3, 4 on the way of receiving energy. And the organism of the patient has a feeling that it needs more and more meal, more food, because we are receiving energy from food. So the next symptom is bulimia. Bulimia. It is eating a lot of meal. Polyphagia, if to say by another word. But all this food, all this meal in the organism, this is carbohydrates, fats and proteins, also producing glucose and ketone bodies. So the organism of the patient becomes something like a plant producing two types of molecules. From all the meal, it is producing glucose and ketone bodies. And this is very dangerous, because high concentration of glucose can cause the comb. It's the symptom when the men are going to die after this. Why this comb appears? Because of high concentration of glucose and influence of high concentration of glucose to the brain. The primary thing is hyperconcentration of glucose in the blood. We say glycemia. glycemia. It is high concentration of glucose in blood. And this high concentration can influence on the brain, causes coma and then death of the patient. And another coma can be caused by the high concentration of ketone bodies, because ketone bodies are acids and pH of the fluids of organism are changing, and this also influences on the function of the brain. And then after ketoacidic coma also can come death to the patient. So this is very dangerous, and these high concentrations of glucose and ketone bodies now can cause coma and death. You understand this? So the main method of treating of diabetes are injections of insulin, because after appearance of insulin, immediately these enzymes appear. Penetration of the membrane for glucose also appears, and after this, concentration of glucose and ketone bodies decrease, and functions of organism can be restored. But except these two comas that is a result of high concentration of glucose, and coma that it is a result of high concentration of ketone bodies, the patient can die from different other complications. And one is very dangerous complications at diabetes mellitus is different infections. Why? Because for bacteria, the organism of the patient with diabetes mellitus is the very good place for life, because bacteria can find much food here, because high concentration of glucose in blood and all tissues is the very good food for bacteria. But normally we are fighting this these pathogenic bacteria with our immune system, and in immune system the most important role belong to immunoglobulin, system of complement, receptors of lymphocytes, and so on. All these substances are proteins. But look, the patient with diabetes mellitus has problems with small amount of energy. So fats and proteins will be degraded and for receiving some energy. Instead of energy, the patient are receiving new and new concentration of glucose, new and new molecules of glucose, and new molecules of ketone bodies. But there is a degradation of proteins in the body of the patient. So very important proteins of immune system, like immunoglobulins, receptors, proteins of complement system are used for producing some energy. So bacteria can eat glucose, and the immune system is not working very good because of low concentration of the proteins of immune system. So finally there are different infections like a pneumonia and other diseases which also can cause the death of the patient. Now we understand how dangerous is this disease, and you also can understand that before discovering the cause of this disease, before discovering the hormone insulin, it was not possible to treat adequately this disease, and all patients with diabetes mellitus died. So now you understand that the discovery of Frederick Banting, discovery of insulin, and obtaining the preparation of solution of insulin, so discovering the method of purification of insulin, saved thousands and thousands human lives. So it is now understandable why Banting received Nobel Prize during 1 year after his discovery. When he was alive, the whole institute, when he was working, was called in his honor. So it was institute named after Banting, and Banting was working in this institute with his name. So it was a great honor for him. And one example I shall show you now. Just after the purification of the first portions of insulin, one woman in northern states of United States of America found suddenly that her daughter is drinking a lot of water and producing a lot of urine. The mother was a medical doctor, and she immediately understood that this is diabetes mellitus, and that her daughter can die very soon. So, of course, it was a great stress for the mother, but she was very happy because she was informed that not very far in Canada, Frederick Banting found the method of treating of diabetes mellitus. So this woman called by telephone Banting, and he said, so you must immediately take your daughter and come to Canada. And the daughter in the train obtained coma. So it was the question of some hours. She could die in some hours very quickly. Fortunately, it was the telephone connection from train to Canada, and they called and said that the girl is dying. So Frederick Banting came to the railway station with the syringe, and it was purified solution of insulin in this syringe. And when the train stopped, he immediately came in the train, in the car of the train, and made an injection to this girl. And girl did not die, because now she obtained insulin. All these enzymes appeared in the organism of the girl, and concentration of glucose and ketone bodies went down. And the most interesting thing, you understood that it is a situation with one of the first patients of Banting. But very interesting thing that mother of the girl. Because she was a medical doctor, she studied the doses of insulin that must be injected to her girl, and finally she discovered the good way of treating of the disease. And the question to you, what do you think? How long this girl lived after the beginning of treating? She, without treating, she must die just in this train. But injection of insulin protected her organism from the death. But how long did she live after this? She lived 60 years more. 60 years more. And she had a lot of events in her long life. She worked, she received many pleasures from the life, and so on. So this discovery of insulin and discovery how to purify insulin from the pancreas saved thousands of lives. And the example with this girl is showing us how important to make such studying of diseases on molecular level. So the future of medicine is the future of molecular medicine, because all diseases will be studied on molecular level, and finally for every disease, I'm sure, will be found the way how to treat the disease, because every disease is the molecular disease. So I hope that today you obtained important information, and I wish you all the best. We will meet with you in the next semester also, and I wish you happiness, and goodbye for today.